Okay, for the very few brave ones that stayed over, and I'm not dying of hypoglycemia yet, I shall be very brief and give you a little bit of a historical review, but more looking to the future, of probably the greatest revolution that medicine is going to undergo, and that's the revolution of personalized medicine. What are the roots of this revolution, and mostly how it's going to evolve, and also what price we are going to pay for it. Because as you all know, there are no free lunches, and we are going to pay a heavy price for it. But nevertheless, we are all walking into it, and willingly and happily so. So the dream of people have always been to remain young forever. And also, the dream of researchers and physicians has been always to cure all diseases. The question is whether this is at all realistic. If you are learning from history, probably this is an unrealistic dream. About 110 years ago, at the turn of the 20th century, as I mentioned in the morning panel, people died at the age of 50. They died mostly of infectious diseases, and nobody has heard at that time, or it was very rare, of cancer, of heart diseases, of neurodegenerative diseases. All these diseases, heart diseases, neurodegenerative diseases, and malignant diseases, came to light only in the 20th century. Why? Because in the 20th century, we almost doubled life expectancy. What happened is that when you are dying at the age of 50 or 45, you don't live long enough in order to get Alzheimer's, or in order to get Parkinson, or in order to get cancer. If you are looking at the curve of malignant diseases, you will see that there are malignant diseases in childhood. But altogether, until the age of 30 maybe, malignant diseases are about 2% of all malignancies and of all diseases. But only at the 4th and 5th and 6th decade of life, malignancies deflect and become a major cause of death. So with age, we are discovering more diseases. So let's try to entertain ourselves that we are going to defeat cancer, and we are going to defeat neurodegeneration, and we are going to live not 80 years like we are living today, but we are going to live 90 and 100 and 110 and 120 years. I'm almost sure that there will be new diseases that are hiding behind the corner that we are still have not discovered yet and have not come to light yet. Therefore, our role now is to fight current diseases rather than to dream on the world that will be disease-free, because for sure, as age is advancing, more and more diseases will be discovered. As I said in the morning, the first 40 years of life are the easier one. The next 40 become more complicated, and I predict that the next 40 will become even more complicated. And there will be the next 40, because life expectancy is increasing all the time. We are now linear, and we are now adding in the Western world about between 4 and 6 weeks of life to every year that is elapsing. That means that about every 10 years, we are adding 1 more year to our life expectancy, all due to advancements in medicine. People that are being born today, in 2012, if the curve will continue to be linear, once they will be at the age of 80, there will be another 10 years almost added to their lifetime. So we are really progressing. Obviously, there is a philosophical question here before we are going to individual, to personalized medicine. And the philosophical question is whether our planet is able to sustain such a population. Remember, 110 years ago, the population of the world was about 2 billion people. Now we are about 7 billion people, and people predict that within less than 100 years, we shall be about 25 billion people. And the economists tell us that this planet is unable to maintain and to sustain more than 20 billion people. That's the upper limit. As far as food resources is concerned, energy consumption, contamination of the atmosphere, and so on and so forth. So very quickly we are approaching the very upper end that economists are predicting, and also environmentalists are predicting the capacity of Earth to sustain us. So not always, and that's philosophically, not always our own interest as individuals and our interest as physicians to cure diseases and our interest as researchers to defeat diseases is in line with the ecology of the planet and with the ability of the planet to sustain it. So there is a kind of a dissonance and maybe even a conflict of interest between our interests as individuals and the interest of the society as a whole and the ability of the planet to really sustain it. So now let's go back to the laboratory bench and see what is ahead of us, nevertheless in medicine, and how medicine is going to change. You are in a medical school, I'm in a medical school, I'm a physician by education, many of you are physicians, become physicians, and let's see what real medicine, what professional medicine is having for us in the future. So let's talk about drug discovery in the 21st century, how it's going to be. But in order to understand drug discovery in the 21st century, we need to go first into history. And the history is not a long history. We are not talking about Roman history. We are not talking about Greek history or Egyptian history. When we are talking about development of drugs, we are already talking about the 20th century. And even not about the entire 20th century, but probably the last 2/3 of the 20th century. Why? There were drugs beforehand. There were drugs in the 19th century, in the 18th century, before. There was traditional Chinese medicine. But these drugs that were developed were never based on scientific grounds. They were kind of hearsay. You know, somebody found something and whispered it to somebody else. They were not science-founded. It's only in the 20th century, with the development of chemistry, that we developed purified compounds and started to make clinical trials, that we were able to communicate the results of experiments to one another and to agree that a certain drug is doing what we are expecting it to do. Not always. We are different genetically, we are different in the environment that we are living. Not on every human being the same amount of drug will have the same effect. If you are trying, for example, to use a beta blocker to reduce blood pressure, for some patients it will reduce the blood pressure more than on others. Some will not respond at all and we will have to change to another drug. But we know the statistics, we know the side effects, and we know that the beta blocker is supposed to relax the tension of the arteries and to reduce blood pressure, to reduce the pace of the heart rate, and then also to reduce blood pressure. We know that insulin is supposed to reduce the blood of glucose by letting glucose go into the cell. On some patients, 2 units of insulin will reduce the glucose by 10 mg/cent. On others it will be 20, on another it will be 30. So we have to titrate patients for the correct response. But always insulin will reduce blood pressure. And we don't need to repeat the experiments. It has been done and tried on millions of patients. And when you are going to the pharmacy and you are buying 10 units of insulin, you know more or less what 10 units of insulin are supposed to do in Brasilia, in Buenos Aires, in New York, in Tokyo, in Madrid, and in Rome. We developed in the 20th century an international language of communicating results of clinical trials to one another. So we know that the drugs are reproducible in their effects and we can trust them within the range of biological response. So let's see what happened to drug development in the 20th century, because we are limiting the whole discussion into probably 40 years that were critically important in the 20th century. And the first revolution was the era of incidental discoveries. And it lasted almost 30 years, between the 30s and the 60s. And let's talk about two major blockbusters that were developed during this time. One of them is aspirin. Aspirin is probably the drug that has been used and still being used mostly in the entire history of the pharma. By weight for sure. Tons and tons of aspirin are being used yearly, and the utilization, the use of aspirin is increased as we talk, all the time. Now, aspirin was discovered completely by serendipity. No research, no intention, no laboratory work, no nothing. The old Egyptians knew already on aspirin. Obviously, they didn't know about the purified chemical. In the willow bark, the Egyptians knew, and it's written, and we have it now all written, that if you chew willow bark, it's very bitter, but it has the potential, the ability to alleviate pain, to reduce pain. The willow barks have a pain-killing characteristic. This was taken for centuries, and nobody really used it until in the middle of the 19th century. A French chemist, Charles Gerhardt, took out the active ingredient. It was salicylic acid, it was very bitter, and he used it to relieve pain. And then it was forgotten again. Until Felix Hoffmann, in the early 20th century, he was a pharmacist that worked in Bayer. Bayer is a pharma company. His father got rheumatoid arthritis. Rheumatoid arthritis is a debilitating disease in which the joints are inflamed, and people are suffering pain, but mostly it's a debilitating disease as far as the movement of the patient is concerned. The patient cannot move their fingers, they cannot button, they cannot use utensils. It also affects the joints of the feet. They can hardly walk, they are basically paralyzed. The father of Felix Hoffmann was a pharmacist in Bayer, got rheumatoid arthritis, you see here the inflamed joints, and he remembered Charles Gerhardt, and he went to his basement to synthesize not salicylic acid, but acetylsalicylic acid, because he realized that by acetylating salicylic acid, he can take the bitterness of the material. So he synthesized the material, gave it to his father, and lo and behold, the father didn't have only the pain gone, but also the disease gone. He basically cured his father from the inflammatory process. And he realized that what he has in hand is not only a painkiller, but also an anti-inflammatory drug. He went to the management of Bayer, convinced them to produce aspirin, and in the early 20s, aspirin came out first to the market as a powder and became a very useful anti-inflammatory and anti-fever and anti-pain drug. Gradually, people discovered numerous other characteristics of aspirin, and there were many Nobel Prizes that didn't go to aspirin, but for the result of what aspirin is doing. And now we know that aspirin is probably the best drug to prevent a second heart attack, and people are also debating whether it's good to prevent the first heart attack. How? By preventing aggregation of platelets and preventing blood coagulation. Aspirin is an excellent drug to prevent blood coagulation, and we know that heart attack is caused by blood coagulation in the coronary artery that leads blood into the muscle of the heart itself. So people that suffer first heart attack are taking aspirin in order to reduce the chances, the risk of a second heart attack, and many physicians like me that have not suffered heart attack take it with the belief that it also can prevent the first heart attack. And there is strong evidence in the literature that aspirin can prevent even the strong heart attack. That doesn't say that aspirin doesn't have side effects. It does have side effects, and people like me that take it just because they want to without any indication, take it at their own risk, but people are free to do whatever they want to do. Then people discovered, and that happened only in the last 10 years, which boosted the use of aspirin enormously, that cancer and inflammation are linked together, and chronic inflammation leads to cancer. Breast cancer, prostate cancer, gastrointestinal cancer, colorectal cancer, and people also realize that from time to time, the inflammation that precedes cancer is subclinical, is subtle. We don't even feel it. We don't have pain, we don't have fever, we don't have anything that will tell us that we have an inflammation. But nevertheless, we have chronic inflammation, and the inflammation at the end will result in malignant transformation. We can discover the inflammation by looking for inflammatory intermediates like interleukin. We should look for them in the blood and in other body fluids, but it's subtle and subclinical. So now it is clear that chronic intake of aspirin can prevent certain types of cancer, for sure colorectal carcinoma, which is probably number 1 or number 2, killing cancer in men and in women, but in women in a little bit later on. So now you can see how from the Egyptian time, aspirin became the most used drug that was ever produced by the farmer. Take another major discovery, penicillin, the first antibiotic. We talked about antibiotics this morning. Complete serendipity of discovery. Complete serendipity. Alexander Fleming, along with Aaron Ciechanover and Florey, received the Nobel Prize for this discovery, but Fleming, the discovery, didn't need to discover. He had no clue what he has in hand. This is for you, this is a petri dish on which bacteria are growing. You can see the white here. And that's how microbiologists are working. By mistake, Sir Alexander Fleming, working in his institute in the United Kingdom, forgot a petri dish open on the table for several days. Instead of covering it and putting it in the refrigerator so to stop the growth of the bacteria, he left it open on the bench and went to a vacation. When he came back, he noted that on the dish, a fungus grew. There was a spore, the air is full of spores of fungi. A fungus spore fell on the rich nutrient dish, and along a few days it grew. Typically, what I would have done in my laboratory, and that happens a lot in every laboratory that grows bacteria, I would have taken the dish and trashed it out. So Alexander Fleming looked at it carefully. And what he saw that around the fungus colony, there is a halo into which bacteria cannot reach. Bacteria grow all the way to here, but they cannot get all the way to here. So he surmised that the fungus secretes an antibiotic material, anti against bios life. And indeed, the first antibiotic was later on, he called Florey and Chain. He was not a chemist, they isolated penicillin, and this was the first antibiotic. Then people say maybe other fungi secrete other antibiotics. Selman Waxman and Albert Schatz discovered another one, which is streptomycin, and the whole world of medicine completely revolutionized. From women that died during delivery, from soldiers and farmers that died out of simple infection, getting a scratch in the field or in the battlefield, we were able to rescue millions and hundreds of millions of people from simple infections that otherwise would have killed them. The problem that we talked today in the panel about antibiotic resistance is something that we see. It's a problem that we should solve, but there is no doubt that antibiotics, again, along with some other fundamental discoveries in medicine, from X-ray to other discoveries, really changed the face of medicine in a dramatic way. So this is antibiotics. So you can see that two of the major blockbusters in medicine came of no research, of no intention, completely by serendipity. But we are out of this era. We probably exhausted it at last. Then came the second era. And the second era we are still living in, and this is the era of high throughput brute force screening. Remember, chemistry really flourished in the last few decades, and people synthesized millions and millions of compounds. Most of them became useless, they are intermediates. Some of them were tried, they became less successful, and at the end we are using a very limited number of compounds. Then people said, oh, we have so many compounds, let's catalog them in libraries of families and use them for something that they didn't meant at the beginning to be used. let's screen them for their potential of becoming a drug. How they can become a drug? Simply, let's think about a keyhole. If we have a keyhole and we forgot the key somewhere, we can bring an expert in. I'm not talking the thief. And the key expert will probably have a collection of 2,000 keys. He will try them one by one. Probably there is a chance that one out of 2,000 or 3,000 keys will open the door. Why? Because the number is limited. The same here. People say we have millions of compounds. We can now grow cells in dishes, and we can model diseases in the dish, or in animals for sure. Let's try these compounds one by one. One of them, by chance, we cannot tell the mechanism. Maybe we'll come to the mechanism later on. But at the beginning, we'll just screen them. And 1 out of millions will stop cell cycle, will stop cell division, and potentially, only potentially, it can become an anti-cancer drug. And people tried and tried and tried, and numerous drugs that we are now used, that are multi-billion-dollar selling drugs, have come to the world by what we call brute screening. Besides the approach, a whole industry developed robots that are going into the refrigerator, take out all these compounds that are all marked by the computer. You should go to the basement of Merck or GlaxoSmithKline. I visited their numerous times, and you will see the huge industry. There are incubators with cells, there are refrigerators with millions of compounds. Robots go into the compound, pull out a tray. The tray is all computerized. They know exactly how many. And then 96 pipettes are going into the tray. 96 drops are going from the tray to the tissue culture dish. The tissue culture dish goes back to the incubator. Then after a day, go to the microscope. The microscope automatically looks at it and tells you exactly which dish there is inhibition of cell growth, or whatever assay, output assay, and the experimentalist has designed for this group of drugs. And then you have leads, you take the leads, you improve the leads by concentration. We have to drop the concentration, so drugs will act not as millimolar or micromolar, but as nanomolar. And then we are going to toxicity, and then we are going to animals and to phase 1. The distance from what we see in the dish to the drug is still huge, but the initial screening for leads start here. Now we are going, why this is not sufficient? I mean, the chances here that at the end we are going to discover drugs to all the diseases. Why people became restless with even the second revolution of medicine? They became restless because there were other drugs that were, just to show you one drug that was discovered by screening, and there are statins. Statins may not be the drug that is sold by tons the most, but certainly by dollars. I can tell you that the 2011 sales of statins, and I'll tell you immediately what are they, has been more than $40 billion, $40 billion by several companies. Statins are inhibitors of cholesterol biosynthesis. They reduce the amount of cholesterol that accumulated in the coronary artery. You see here the coronary arteries, for those of you who forgot or don't know, are the arteries that take blood to the heart itself. Once the cholesterol accumulates, then there is blood clotting on top of it, and we are occluding blood flow via the coronary artery to the heart, and we are ending up with a heart attack. Now we are able to reduce the amount of cholesterol by inhibiting the synthesis of cholesterol by statins. Statins are consumed by millions and millions of people worldwide. They were discovered initially by Akira Endo, a pharmacologist in a Japanese company. He was awarded several years ago the Lasker Award, which is one of the most prestigious awards given to biomedical researchers. And it was discovered by Akira by putting an assay for the synthesis of cholesterol, taking the whole library of natural compounds, several thousands of them, and screening them one by one, until he found the one that inhibited. No mechanism behind, no structural biology behind, no nothing behind. Just brute force screening that led to the discovery of probably one of the most important blockbusters and not only by dollar making, but also by life-saving and morbidity-saving drugs that history has ever known. Let's go to the last revolution, and that's the revolution of personalized medicine. What was wrong with the first two approaches? The serendipity, we understand that we cannot rely on it. We cannot rely on serendipity. But what's wrong with continued screening? We should screen and screen and screen and screen until we should eradicate all diseases. What's wrong about it is that the diseases are not the same, though we think they are the same. And let me explain it. We have here a group of patients that are diagnosed today with a certain disease. Let's say that this is a group of women that have breast cancer, or a group of men that have prostate cancer, or a group of people that have Alzheimer's disease. We are starting to treat them, and then we are following them. And what will happen when we follow them, following 5 years, we shall see that some of them, the green ones, were cured. But the blue ones and the yellow ones died. How is it that a group of women that looks the same, all are around 50 years old, that have a tumor in the breast that didn't metastasize yet, they started the same treatment, and within 5 years, they are taking a road that is completely and vastly different from one another. This woman died in suffering from a devastating, stampeding, metastasizing disease that didn't respond to surgery and to chemotherapy and to radiotherapy. While these women are back to their family healthy and happy. And the same for the man with the prostate cancer. Why is it that for some people the disease is going very, very slowly, and for some people the disease is going very, very fast? Because it's not the same disease. We are different genetically, we are different environmentally, we are different dietetically, we are different by any means that we can imagine. And therefore, we respond different, and the disease that is evolving in each and every one of us, despite being defined as the same disease. Oh, this woman has a breast cancer, and this woman has a breast cancer, and this woman has a breast cancer. The three women may have a different breast cancer, and therefore they respond differently. And we need to understand why. And in order to understand why, we have to profile the woman and the man genetically for the causes of their disease. And that's what we are doing now. And in order to do that, we are entering a new era that is called the 4 P's medicine. It's not only 1 P, but it's 4 P's. It's personalized, it's predictive, it's preventive, and it's participatory. And these are the terms, not of me, of Ehud, who is the founding father of this concept. Now we are already in this revolution. It's not that one day in 12 years we will wake up and read in the New York Times that as of this morning, or as of January 1st, 2041, the revolution started. This is not a war that erupts in the morning. We are already in it, but in a mild way. But we are going to enter it in a much more stormy and strong way as time passes. And I want to give you one example. Look at the biopsy that is taken from the tumor of 2 women breast cancer. These 2 women, woman A and woman B, has breast cancer. But you can already tell that this woman is different from this one, because I can stain the tissue of this woman for something that I cannot stain the tissue of this one. What is it? This woman has a mutated estrogen receptor. The cancer in this woman is driven by a mutated estrogen receptor. Therefore, this woman can be treated by a tamoxifen that is an anti-mutated estrogen receptor. While treating this woman with tamoxifen will be useless and helpless and wasteful. She will waste the money of the drug. The drug will implant hopes in her that she is being treated, and basically we are giving her water, nothing more. For this woman, I need something else. Maybe she has a mutated estrogen progesterone receptor, maybe she has a mutated EGF receptor, and maybe she has something else that I don't know about. So already you can see that I start to stratify. 2 women with breast cancer, one has breast cancer A, and one has breast cancer B. And here is the woman that has breast cancer B. This woman has a mutated EGF receptor, and therefore she can be treated with a drug that is called Herceptin. But only a woman that has mutated EGF receptor can be treated with Herceptin. Yes, the other ones can also be treated with Herceptin, but it will be like giving them water. So now we have already two women with two different breast cancer, mutated EGF receptor and mutated estrogen receptor. And I can tell you that there is another woman with a mutated progesterone receptor. And then there is a fourth woman that is called triple negative. She doesn't have one, or the second, or the third. And she is the most aggressive and the most risky one, because we cannot offer her anything. We have nothing for her. The only thing that we have for her is a hope that we shall discover the reason of why she has a breast cancer. And we are working on it, not we, the scientific community. How? By making whole DNA genome sequencing. By taking the whole DNA and trying to find new mutations that drive breast cancer. And we are doing it, the Broad Institute, the Human Genome Project, there are many projects that are running in parallel all over the world. They are doing it by looking, by sequencing thousands and thousands of samples of the same disease. By taking, let's say, 10,000 women with breast cancer, taking a sample of their DNA and sequencing it, we hope to be able to get a roadmap of a map, classifying them into a group that has a mutation in A, a mutation in B, a mutation in C, D, E, F, and G. So from now on, in the future, there will not be any more breast cancer. There will be breast cancer A, B, C, D, E, and F, and G. In other words, the mirror of medicine is going to turn 180 degrees from the disease to the patient. The first thing that we are going to do when the patient is coming to the hospital, even if the woman has a breast cancer, is profiling the patient DNA. We are not going to treat the tumor first. We are going first to diagnose the type of the tumor and only then treating it. So the whole concept of medicine is going to be very different. And it's not going to be only, I have to shorten it because of time and constraint. We are not going also not only to profile DNA, because we know DNA is not the cause of the disease. People say, you have a mutation, you have a disease. At the end of the day, what causes diseases are proteins, not DNA. So we have to look at all layers. We are looking at the RNA level, we are looking at proteins, we are looking at proteomics, and we are looking also at dynamic proteomics, because proteins are undergoing modifications that are not encoded in the DNA. They are going post-translational modifications, from phosphorylation to acetylation to amidation, glycosylation, ubiquitation, you just name it. And all those changes, first of all, the mutations in the coding sequence, but all those modifications may well be involved in pathogenesis of diseases, and we need to understand it. So we are going to genomics, we are going to transcriptomics, to RNA analysis, we are going to proteomics, and we are going to post-translational proteomics, and then we are going to metabolomics, because we want to understand also the products of the enzymes that are there. So you see that medicine, from being very limited these days to certain parameters that we are monitoring, is going to expand exponentially in the amount of information that will be available for us. For now, we are monitoring all kinds of parameters. Think about biochemistry today. For those of you who are students of medicine or internists, you are opening Cecil or Harrison textbook, and at the end, you find a table of data that will tell you about 200, 300, 400 biochemical parameters. This is going to be nothing. We are going to face an avalanche of thousands and thousands of thousands of different parameters that are going to be changing during diseases. Obviously, there will be computers that will be able to classify them and to help us reach a diagnosis. Let me come to the very last slide. I've really shortened my talk. And to talk to you now, very briefly, about the difficulties and the price of this revolution. Not that these difficulties are going to deter us, on the contrary, We are going to overcome them. But just to show you that just going into the DNA and finding one mutation is not the story. The story is much more complicated. So, many diseases, mostly metabolic and psychiatric. You know, in society we still don't talk on psychiatric diseases. But let me tell you, the psychiatric diseases are the most common and the most burdening diseases that we have ever met in medicine. The amount of people that are suffering from depression is daunting, is unbelievable. And the burden of these diseases on the economy is huge. Because these people, many of them are not sick in the simple way of the term that they call work and say, oh, I'm sick today, I'm not coming. They do come to work, they function, but they function only 20% of their capacity, or 30%. So think about working power, think about efficiency, think about what happens to society when 10% of the people are defined psychologically as suffering from depression. All of these diseases, and many metabolic diseases, are multigenic. So there is not a single gene. So once we are going to identify the genes, we should find that these diseases are caused by 10 or 20 genes. And then each gene is penetrated to a different level in a different patient. The expression of the gene in the different patients is different. So to understand and to assess the contribution of each gene in a group is much more difficult than to assess the contribution of a single gene. When we have 10 or 20 genes that are all causing the one disease, it's very difficult to assess how much each is contributing, especially that the level of expression of these genes in different patients is different. Then, malignancies are characterized by genomic instability. We start with one mutation, and we treat the patient, and the patient responds to the treatment, to the chemotherapy or radiotherapy, and then all of a sudden the patient evades the treatment. Why? Because the patient developed a second mutation, and a third mutation, and a fourth mutation, and a fifth mutation. The genome of cancer patient is unstable. So we need to follow the genome as we progress with the disease and change the treatment all the time. But changing the treatment we can say, but we don't have a repertoire of drugs to tackle mutations in all the potential genes. The repertoire of drugs is limited. So the fact that the genome is changing at the end will lead to the death of the patient, because at a certain point the oncologists say, that's it, I exhausted all my arsenal. Now human experimentation is very complicated and very expensive, and we cannot do experiments on human as we wish, but at the end we need to make experiments on human beings. Because we can cure mice from cancer, but the same drug that will cure the mice will not cure human beings. Why do not cure human beings? Because human beings are not mice. Mice are belonging to one species, they are very limited genetically, they grow in cages, the light is turning off at 8 o'clock in the morning, turning off at 8 o'clock in the evening. They eat chalcorina, which is identical to all the mice. They are not traveling, they are not exposed to irradiation. Mice are mice. We learned already this bitter lesson, that we developed hundreds of drugs, if not more, that could cure cancer of mice. None of them was even starting to be effective in human cancer. So we learned it the hard way. Obviously, we lack faithful animal models. Think about neurodegeneration. Think about diseases that are evolving along decades. You know, Parkinson started at the age of 20. We diagnose it at the age of 50 or 60 because it takes 30 or 40 years to degenerate all the substantia nigra cells, par compacta, the substantia nigra par compacta in the brain. But we cannot wait with animals. You know, animals are living, so even the mouse is living 24 months, 27 months, they don't develop these diseases. And what about depression? How can we tell that the mouse is depressing? We can ask him to dance, to tell a joke, to smile, to do something. Schizophrenia. How can we assess schizophrenia in mice? Mice are mice, rats are rats, and human beings are far more complicated. We should remember it. And then many of the drug companies are liable today to all kinds of lawsuits. I don't want to go into it. And as I said in the morning in the panel, they are reluctant to develop certain drugs. They are reluctant to develop antibiotics. They are reluctant to develop any drug that has a risk in it. And I shall not tell you what drug companies, how they are weighing and how they are considering whether they are entering development of a certain drug or not. But let me just tell you the bottom line. Our fate as human beings living on this planet is being put at the board of directors of private companies that are interested in increasing the value of the share of their companies. So think about this huge philosophical dissonance between sanctity of life and what we are reared and educated as physicians and as human beings, and what we are swearing when we are getting our diploma at our commencement to the interest of the pharma industry. Last but not least, I put it last, but probably one of the most important considerations are bioethical problems of availability of genetic information. Think about, we are depositing now my DNA in the hand of somebody, my physician, that now in 10 minutes will have the ability to sequence it and to find all my mutations. Now let me just give you 2 or 3 short examples of the potential risk of this. I'm not talking about insurance companies, that the information can lead to the insurance company and the information we know that in 5 years I should develop cancer, and once I will be treated for the cancer I should be depressed, and then following it I should have an heart attack. And so no insurance company will ever want to insure me despite the fact that currently I'm healthy. So think, this is simple, forget about it. But think about the doctor is doing sequencing and he discovers there a gene that makes him susceptible for Alzheimer's. And currently there is no treatment for Alzheimer's. First of all, to whom this information belongs? Does it belong to the doctor that found it? Or does it belong to me that I am the source of the DNA? Let's say that the doctor thinks that he wants to tell me what's in my DNA. What if he will help me? Now the doctor knows something about me that I don't know. Now he will come and tell me that in 10 years I'm going to have Alzheimer's disease, or schizophrenia, or whatever, a disease that has no treatment. How my life are going to look like after I'm being told about something that anyway medicine cannot help with. So, and let me now tell you the last example that will really show you how complicated it is. A woman of 50 years old was operated yesterday for breast cancer. The breast cancer was taken away, she doesn't have metastasis to the lymph node, she's basically cured. The doctor tells her, you know, there is another genetic test that I want to do on you for breast cancer susceptibility. And the genetic test is on a gene that we know already that carries high susceptibility for breast cancer, and it's called BRCA1, BRCA1. Women that carry this gene are highly susceptible to breast cancer. The woman will tell me, what do I care? You took my breast away anyway. I had breast cancer. The doctor will tell her, yes, but you have another breast. And the other breast is not immune. She undergoes the test, and she discovered to be positive for a mutation in the gene. And she's 50 years old, she has 3 daughters, and she says, what do I care? Let's take the second breast away also. The operation is much more complicated. I don't want to go with you to the operation because we need to take all the breasts. I'm coming from the world of surgery, so I can tell you. I'm a surgeon by training. So the operation is to take all the breasts out, all the breast tissue, not only the tumor, but forget about it. She agrees to take the second breast away. Then the doctor tells her, well, but this gene also carries susceptibility for ovarian cancer. And ovarian cancer is a deadly cancer because most of the women that have ovarian cancer are dying because it develops in the abdomen and it's late to be diagnosed and it's metastasizing and so on. Wow, a new story. So a woman that started yesterday with a single small tumor in the breast, ending up taking her second breast away, and also now she said, okay, I have 3 daughters, I don't need my ovaries, I should take hormone therapy, let's take the ovaries out as well. This is already complicated. Now start the real problem. She has not even started to undergo to solve her problem. This woman has 3 daughters. One is in high school, 17 years old. The other one is 21, just starting college, and the other one is 26, married and has 1 child of 2 years old. I'm telling you a true story. The gene that she has may have been transmitted to the daughters. Now what she is telling the daughters about carrying a gene that has a high susceptibility, a really high susceptibility for breast and ovarian cancer. What the daughters are going to do? To take the breast away, to take the ovaries away, they don't have children, to go every half a year to the doctor to see whether they have cancer. What the daughters are going to tell their boyfriend or the husband, that the husband or the boyfriend is walking with a ticking bomb, and the ticking bomb may blow out every day in his face. Just imagine a simple case of a mutation and BRCA1. This is not a favor, this is not imagination, this is a mutation in a gene, and we know exactly the percentage of women that are carrying mutation in BRCA1 gene. So, with knowledge come troubles, but science should know how to tackle them. I don't have the time to go into, I wouldn't say potential solutions. We don't have any potential solutions about even ideas raised in discussions and who are the discussants and who participate in such. But I can tell you that medicine is going now willingly and happily into probably the most important revolution it has ever undergone, that will change completely the face of medicine and the face of every patient with enormous potential, but at the same time with an enormous risk and problems side by side. So thank you very much for your